Salve, buongiorno a tutti. È un mio grandissimo piacere, nonché onore, introdurvi il professor Di Cera, che è nato a Palermo, conseguito la laurea in medicina e chirurgia presso l'Università Cattolica del Sacro Cuore a Roma. Poi ha lavorato come postdoc all'Università del Colorado con Stanley Gill e Jeffrey Wyman. Nel 90 è diventato docente presso la Scuola di Medicina della Washington University a St. Louis e nel 2010 si è trasferito presso la Scuola di Medicina della St. Louis University come professore chairman del Dipartimento di Biochimica e Biologia Molecolare. Il professor Di Cera è anche membro della National Academy of Inventors e dell'American Association for the Advancement of Science, autore di più di 300 pubblicazioni su riviste internazionali di grande prestigio, è un esperto di criomicroscopia elettronica e la sua ricerca è basata sull'enzimologia strutturale dei fattori della coagulazione, che è finanziata da più di 30 anni dall'NIH. E fatemi dire anche che ho apprezzato, da quando lo conosco pochi mesi, un'innata disponibilità e umiltà che sono virtù che stanno sempre più difficili da ritrovare. Grazie.
Ok, questo è in inglese chiaramente il seminario. No, dice che bisogna farlo in inglese. Okay, so thank you very much for the introduction. Claudia, it's better if we go English because I forgot a lot of Italians. I wouldn't know how to translate folding in Italian. How do you say folding in Italian? Ripiegamento. Impacchettamento delle proteine, ripiegamento. Yeah, that's not a good way to translate folding. So if you don't mind, I will speak English, and also if there are people connected via Zoom, they don't speak Italian, we'll appreciate that. So I really appreciate the invitation. Thank you very much. I apologize that we had to move the seminar from Monday to to today. Unfortunately, that's not my fault. They canceled my flight, and I arrived in Rome on Monday when I was supposed to be here for the seminar. So anyway, what I would like to do is to talk about the structural biology of coagulation factor V, and I want to start with a slide that I've stolen from a recent review published by the group of Professors Sika, whose work I follow for many, many years, and first author is Troisi. And what I liked about this slide here, this figure in the review, is that for the first time somebody took the effort to give a depiction of the coagulation cascade, which is a mess, okay, in terms of the structural biology. And so for all the elements constituting the coagulation cascade, in this slide you see basically all the players with what's known in terms of the structure, whether it's an X-ray structure here in brown, a cryo-EM structure in green, or no structure at all, a prediction from AlphaFold in blue. And what you see is that we know quite a bit. There is a lot more to know, but some of these proteins have been characterized structurally. And so we can, for the first time, really look at the players of this complicated transduction mechanism. That's exactly where it is in terms of the structural biology. And today, we're going to focus on the properties of coagulation factor V. But you can still appreciate, even though there are beautiful pictures in this slide, this is complicated. There are two pathways, the extrinsic pathways, the intrinsic pathways that converge to a common pathway to activate thrombin at the bottom of the cascade that will then go on, cleave fibrinogen and form a fibrin clot. But it's kind of difficult to follow these things. And this is the reason I think that many people, when they're supposed to do coagulation cascade, they say, no, I want to do something completely different. This is too much for me. So I'm going to give you a version of the coagulation cascade, which is the way I understand it, which is very simple. Okay? There are two pathways of the coagulation cascade. One is called the extrinsic pathway. This is the pathway that's activated if you cut yourself accidentally and produce a lesion of the vasculature. This extrinsic pathway, it's called extrinsic because it's triggered by tissue factor, which is a protein not normally present inside the blood vessel. Is exposed when you cut the endothelium, and it binds to factor 7 and activates this pathway. There is an alternative way to activate the coagulation cascade. It doesn't require a lesion of the vascular wall, but something that alters the nature of the vascular wall. And this happens especially in pathological conditions that will lead to myocardial infarction and stroke. And so this is called the intrinsic pathway because it's mediated by proteins that are normally present inside the blood vessels. But both the extrinsic pathway and the intrinsic pathway then converge to a common pathway where basically a coagulation factor called factor 10 is activated to factor 10A and cleaves a very small amount of prothrombin or coagulation factor 2 into thrombin. It's a very, very small amount of thrombin, which is generated at the beginning of the reaction and when the coagulation cascade is triggered. But when thrombin is activated, even in minuscule amounts, it can activate this coagulation factor 5 that we're going to focus on. And the activated form of coagulation factor 5, called factor 5A, then will bind to 10A and form a very powerful enzymatic complex called the prothrombinase that now increases the specificity toward prothrombin by 10,000 times, assembled on the surface of platelets. And at this point is the massive generation of thrombin, explosive generation of thrombin, that really triggers the formation of a blood clot, the aggregation of platelets, and also inflammatory response. So this would be the final step in the coagulation cascade where it's either activated by the intrinsic or the extrinsic pathway, and it's mediated basically by the action of thrombin on the activation of minuscule amount of factor 5 into factor 5A activated. And so factor 5A is a powerful cofactor in the prothrombinase complex that is responsible for the massive generation of thrombin from prothrombin. But as you trigger the coagulation response, you also have to figure out a mechanism to inhibit this response. Otherwise, you will clot the entire amount of fibrinogen you have in the blood. It will turn into a candy. And so what happens at the level of capillaries where the formation of blood clot would be extremely dangerous because it would block the blood flow and cause complete blockage basically of the circulation. Thrombin binds to a receptor on the endothelium called thrombomodulin, which is here TM. And when bound to thrombomodulin, thrombin can no longer cleave fibrinogen, or activate platelets, but it can activate a protein called protein C. And the activated form of protein C or activated protein C with the assistance of the anticoagulant protein S, and we're going to talk about this in a little bit, will inactivate factor 5A. And inactivation of factor 5A will shut down thrombin generation and formation of further blood clot and platelet aggregation. So basically, at the basis of the procoagulant and anticoagulant functions that you have in the blood is coagulation factor 5 and its activated form. And basically, the pathology that's associated with it is that if you have defects of factor 5, you will have bleeding, and if you have defects of factor 5A, so if you could not inactivate factor 5A with the activated protein C and protein S axis, you will have thrombosis. So this is, in a simple diagram, really recapitulations of what the coagulation cascade is all about. And at the center of the control, or either the procoagulant or the anticoagulant activity of the coagulation cascade, is coagulation factor 5. And we're going to talk about factor 5, and we're going to show some results that we obtained recently with cryo-EM. And you may want to ask, why do you want to use cryo-EM instead of X-ray crystallography to study coagulation factors? Because coagulation factors are very large. They're too large for NMR analysis. NMR has been applied to thrombin in some cases, but thrombin is very small and not comparable to some of the proteins I'm going to talk about. And the other advantage of cryo-EM, it will capture these proteins in a native state that is unlike what you have in X-ray crystallography, where you have to align all the proteins in the crystal exactly the same way to have efficient diffraction. And this technique of cryo-EM is particularly suited for coagulation factors, as I said, because they're too big for NMR. And in some cases, in many cases, actually, I should say, they're very, very difficult to crystallize. Using cryo-EM, we've been successful in crystallizing several of the coagulation factors and their complexes, shown here, summarized. There is another one that's coming up soon. And we're going to focus for the rest of the talk on coagulation factor 5. So coagulation factor 5 was discovered in 1944 by Paul Oren. And when Dr. Oren discovered factor 5, he was a very young, totally unknown assistant professor. Nobody believed him. And it took the field almost 10 years to realize that factor 5 was a truly important coagulation factor. So the reason I say that is because there are people in the room that are young assistant professors. If you think you made an important discovery, nobody believes you, don't worry about it. Stay the course, you're in very good company. So Dr. Oren took almost 10 years to convince everybody that coagulation factor 5 was very important. And the reason why he called it coagulation factor 5, because at the time there were only 4 coagulation factors known. There were tissue factor, prothrombin, fibrinogen, and calcium. And so he didn't come up, we didn't have a very good name for the factor, just call it factor 5. And he used a Roman numeral to number it, but that has the profound impacts, because after factor 5, all the coagulation factors were named after the Roman numerals. And tissue factor, prothrombin, fibrinogen, and calcium were also renamed with coagulation factors 1 through 4. So coagulation factor 5 is a very interesting molecule because it acts as a procoagulant, as a precursor of factor 5A. And as I said, 5A is an important cofactor of prothrombinase that's responsible for the generation of thrombin. But it paradoxically also acts as an anticoagulant because it's the target of APC inactivation of factor 5A, and is also a cofactor of the APC inactivation of another coagulation factor, which is coagulation factor 8A, which is involved in hemophilia. And that reaction involves the intervention of the anticoagulant protein S. Factor 5 is a very large protein composed of over 2,000 amino acids arranged in 6 domains, 2A domains, 3A domains, 2 C domains, and a very large B domain. It's activated by thrombin upon cleavage at arginine 709 and 1545 that removes completely the large B domain, generates a 2-chain molecule held together by calcium. It is in this form that the activated form of coagulation factor 5A binds to 10A in the prothrombinase complex. So the structure of factor 5 was solved by cryo-EM by a postdoc in my lab, Eliza Rubin, I guess now 5 years ago. It's been a long time. And it shows the arrangement with the 2 C domains at the bottom, here forming a membrane binding module and supporting on their top the A1 and A3 domain disposed side by side. There was nothing wrong with this arrangement because it was already reported in a pioneering X-ray structure of the bovine inactivated form of factor 5A that was solved by Eversey and Mann many years ago. But what was new about the cryo-EM structures solved by ELISA was the identification of the A2 domain. And with that, of regions important, as we will see for the activation of prothrombin that we call the lid and the gate, and the sites of APC inactivation are arginine 506, which is mutated in factor 5 Leiden, and the arginine 306, which is mutated in factor 5 Cambridge and Hong Kong. And of course, the two sites of activation by thrombin, and arginine 709 and arginine 1545. But what was very disappointing about this structure, if we can say such a thing, was that when we looked for the large B domain composed of over 800 residues, we could only locate about 4 residues at the amino terminus of that domain and 10 residues at the C terminus. So we were missing a lot of the real estate of this B domain. And this was disappointing because the B domain plays very important roles in factor 5. First of all, it screens arginine 306 and 506 from inactivation by activated protein C, and also orders both the A2 and A3 domain. And this was shown very convincingly in the structure that ELISA solved of factor 5A, the activated form of factor 5, that missed completely almost 100 residues in the A2 domain and 37 in the A3 domain, and got rid of all the information about the lid and the gate that we saw in the inactive form of the cofactor. And so that's entirely, this entire piece of the molecule was gone, could not be located due to the disorder of the B domain. But interestingly, when you bound factor 10A in the prothrombinase complex that was also solved by ELISA, these regions came back to life and could be located in the cryo-EM map. So there are portions of the 5A molecule that are intrinsically disordered. They become ordered upon binding to biological partners. But there is another reason why the B domain is very, very important, because it's the domain that keeps factor 5 in the inactive state. And that mechanism is an intramolecular interactions between a basic region toward the end terminals of the B domain and an acidic region located almost 500 residues downstreams that come together intramolecularly and keep the molecule in its inactive form. So without a structural understanding of the nature of the B domain, it becomes very impossible to say what is the nature of this intramolecular interaction, and if indeed this is the mechanism that keeps factor 5 in the inactive form. So where is the B domain? What's the structure of the B domain? And these days, when you don't know the answer to a structural question, you ask AlphaFold. And AlphaFold, I must tell you, works extremely well in some cases. In the case I want to show you is the case of the anticoagulant protein S. Anticoagulant protein S is a cofactor required for the inactivation of factor 8A by APC and factor 5. So it's a very important molecule. The structure of this molecule is currently unknown. And what you see here in the cartoon representation is what AlphaFold predicts as the structure of protein S. And that prediction is spot on the molecular surface that we determined recently by cryo-EM. So they're almost identical. In fact, the prediction from AlphaFold is so good that we're using that as a model to refine the final structure of protein S. But we wanted to really verify that things were rigorous and in an independent manner. And so we used a very powerful new technique called quantum-enabled pulse EPR spectrometry, through which you put some spin labels to cysteine residues that you have to engineer in the protein, and you can measure distance. And the distance you measure is extremely accurate, more accurate than if you use, for instance, single molecule FRET because the probes are very, very small. And we put 2 probes in the EGF2 domain of protein S and the laminin, second laminin domain. And the predicted distance, according to the AlphaFold model and our preliminary cryo-EM structure, is about 48 angstrom. And when we measure with the quantum EPR, the dynamics and the distance between these 2 probes put in the EGF2 and the laminin 2 domains, what we found was very interesting. Were 2 populations. A larger population, 91%, was 46 angstrom, practically identical with the distance predicted by the AlphaFold and our cryo-EM preliminary structure. And then a second population, much smaller, about 10%, that suggested there will be a contraction of this distance as though the protein S will kneel using the EGF2 as an hinge point. And interestingly, very recently, we found that we have particles that not only reproduce this envelope here that I showed you, but also a second class of particles where there is a compactification of the structure is more consistent with this 23 angstrom resolution. So you know now where we want to go with factor 5. We're going to use basically the same strategy. Attach the probes, measure the distance, and see what that says about the B domain. So we started asking AlphaFold for a predictions of the B domain of factor 5, actually the entire factor 5, and what we got was this. Basically the body of the protein, which is here in different colors and in surface representation, is pretty much the same as the structure that we have solved. So AlphaFold is very good at predicting the structure if you solve the structure already, because it pulls the structure from the PDB and says this is the predicted form that you're looking for. But when it came to the B domain, what the prediction was, according to AlphaFold, was this spaghetti-like structure, which is basically a way for AlphaFold to say, sorry, I have no idea what this domain looks like. But notwithstanding this uncertainty, you see the B region, which is the basic region in blue, and the acidic region in red, they come reasonably close together in space, suggesting that indeed there is a tendency for these 2 domains to interact. But we couldn't get any information from this prediction where to put probes to do the EPR experiment. And at this point, a postdoc in my lab said, maybe we forgot something very important about factor 5, and that is that factor 5 has a lot of post-translational modifications. So it's glycosylated, it's sulfated, and what if we introduce these sites of post-translational modification according to the biochemistry and the database? Is the prediction from AlphaFold any different? And indeed, the prediction is completely different. Now, instead of a spaghetti-like type of structure, you get a superstructure of alpha helices, all in the front of the molecule. So this would be the predicted B domain according to AlphaFold, based on the KJ, which is Kevin Jagesar initials of my postdoc in the lab. So this would be the predicted structure, and very interestingly, the B region and the A region come very close to each other. You can identify contacts that you can target by mutagenesis on top of the A2 domain. So which prediction is correct? Is this prediction that will explain why it's so difficult to capture this by cryo-EM? If the B domain is indeed very unstructured, as the spaghetti-like prediction would say, that will explain why you cannot see it by cryo-EM. But on the other hand, this superstructure will explain the biochemistry. It will tell you that the BR and the AR come together, keep the B domain in a conformation where the factor 5 will be inactive. But the thing we don't understand is if this is indeed the structure of the B domain, how come we do not see it by cryo-EM? If it's so structured, why is it that we don't see it? And so we looked at whether this prediction will explain the biochemistry and other things. And so here I've rendered the B domain surface representation, and you see how massive it is in front of the rest of the molecule. That's it's in the background here. And in this position, it will completely cover the epitopes that are necessary for factor 5A, the activated form of factor 5, to interact with prothrombin and 10A in the prothrombin-prothrombinase complex, or even with the activated form of protein C that will attack arginine 506 and inactivate it. And in fact, if this is the structure of the B domain, that will explain beautifully the biochemistry and why the factor 5 in the inactive form will not be active in the prothrombinase complex. It will not be able to bind prothrombin and 10A because they will have to occupy exactly the same epitopes of the B domain. But it will also explain why APC it will not work for the inactivation of factor 5. But most compelling for us has been the collection of new data on factor 5, where now we got to see some extra density in front of the A2 domain. And this extra density sits right where the interaction between the B region and the A region in the predicted model of KJ will sit. So we're pretty confident we're starting to see pieces of the B domain that will reveal the origin of the mechanism of the inactive form. Other things that are interesting about this structure is that the B domain in this conformation will not prevent the C domains to interact with membranes, so it will not prevent factor 5 to start getting activated by thrombin and sitting on a membrane surface to cleave prothrombin. And and on the other hand, it will leave the back of the molecule free to interact with other components of the blood coagulation, and especially the protein S and factor 8A to explain its anticoagulant role. So we are pretty sure that this predicted structure of the B domain is very close to being the real thing, and the reason why we have not been able to detect it by cryo-EM may be due to the fact that it's very dynamic, and so by optimizing conditions with the grid and maybe having more powerful cameras on the microscope, which is also what made these extra measurements possible, will eventually reveal the entire structure. So, but the question is, what is the B domain? What does it look like when you can actually see it? And there is a derivative of factor 5 called factor 5 short, which is a splice variant, has a shorter B domain, 133 amino acid instead of 836, and it contains the sites of activation by thrombin at 709, 1545, and the sites of inactivation by APC. But with the shorter B domain, it has no basic region, it has the same acidic region, and so it acts as a procoagulant because not having the mechanism that inhibits intrinsically the factor 5, it will have the acidic region free to interact with 10A and cleave prothrombin in the prothrombinase complex. So that would explain the procoagulant activity, factor 5A-like or factor 5 short, and also why the C-terminal end of TFPI alpha, which is another inhibitor in the blood coagulation that is highly homologous to the basic region of factor 5, can interact with the acidic region, and then the complex formed by 5 short TFPI alpha protein S and will hijack coagulation factor 10A and form a complex that is anticoagulant and explain the associated bleeding disorder with the so-called East Texas syndrome. So that's a very important derivative of factor 5 that's present in the blood of all individuals, including normal individuals, but is present in larger amounts in patients affected by East Texas disorder and suffering bleeding disorders. So the B domain, factor 5 short, is much shorter, but retains the acidic region, and a region that precedes the acidic region, this pre-AR, is also very important. So we managed a few years ago, 2 years ago, to solve the structure, Bas and Mohammed solved the structure of factor V short by cryo-EM, and he was able to see completely the B domain from arginine 709 all the way up to the other side of the molecule, up to the splice region near arginine 1545. Then the B domain will continue to form an ascending arm where the pre-AR is located, and the hydrophobic patch identified by Dalbach as a site of interaction with protein S, anti-FBI alpha, and then it will continue toward the top of the A2 domain here to define the acidic region. And the acidic region will then terminate at arginine 1545 at the end of the B domain. And the ability to solve now the structure of a derivative of factor V in its complete form gave us an opportunity to define the electrostatics associated with it. And as you can see here, in the top of the region comprising the A2 domain and the AR has a very large negatively electrostatic surface that we believe is the locale for the interaction with the FBI alpha, that region, and that will recruit protein S right toward the back of factor V short, and then factor 10A will bind to the front of the molecule. And we're making progress in solving the structure of this quaternary complex, and we see that pretty much this theoretical arrangement based on the electrostatics is pretty much correct. So why is the B domain of phi short relevant to factor V? Well, because last year, Bassem was able to refine additional particles of the original set that led to the solution of the structure of factor V, and identify additional residues in the acidic region, which are labeled here in cyan. Instead of just 10, it went up to additional 25 residues. And you can see that the acidic region climbs on top toward the A2, on top of the A3, and toward the A2 domain, just like we saw in factor V short. And then it connects up to this additional acidic region, which is the 660, 668 domain of factor V. And the important thing in this analysis of factor V is that the structure of the acidic region of factor V, now in this a new set of structure, which is here in cyan, matches almost identically the conformation of the same region factor V short. And the two regions actually agree very well with the predicted structure from AlphaFold. Actually, I should say the other way around, that AlphaFold makes a prediction which is actually very much in line with the current structures of the factor V acidic region and the factor V short acidic region. So you see the overlap between the red, green, and cyan in these slides. So that's pretty much where we are with the B domain. We're continuing our analysis, and we think that pretty soon we will get a solution for this puzzle and being able to determine the architecture of what keeps factor V in its inactive form. And then in the last part of my talk, I want to switch gears a little bit and tell you about the procoagulant role of factor V and role in the prothrombinase complex. And we're going to talk about the activation of prothrombin. Prothrombin is a multi-domain protein composed of a GLA domain, here 2 Kringle domains, and a protease domain. And its activation to thrombin can follow two pathways. One is called the prethrombin 2 pathway, where prothrombin is first cut at arginine 271 and remains just with the protease domain. That domain is then cleaved at arginine 320 and activated to thrombin. And the alternative pathway is when you first cut at 320 and generate an active intermediate called mesothrombin, and then you cut mesothrombin 271, get rid of the auxiliary domains, the GLA domains, and the 2 Kringle domains, and get back to thrombin. So these two pathways are context dependent. On the surface of platelets, this prothrombin 2 pathway predominates. On the surface of the endothelium is the mesothrombin pathway that predominates. But in vitro, if you use factor 5A, basically you select the mesothrombin pathway. So if you use 5A in vitro, you always activate prothrombin along this pathway. First generate mesothrombin, and then you generate thrombin. So 5A is extremely important, at least under the conditions that you can study in vitro, in determining the preference between the two sites of cleavage that are necessary to convert prothrombin into thrombin. And so we solved the structure of prothrombin bound to prothrombinase, and we saw, first of all, that the prothrombinase complex, the 10A, this is 5A, this is 10A in the front, has the same conformation whether you have nanodiscs or not, whether you solve the structure on a nanodisc or in solution, the structure is pretty much the same. So the role of the membranes is not to change the conformation of the proteins and make it more active, but rather to just facilitate their encounter and reduce the dimensionality of the system and increase the diffusion. In this complex, you see 10A that aligns along the C1, A3, A2 domains of factor 5A, and prothrombin aligns along the C2, A1, A2 domains. And they face each other, interacts only through their protease domain. And then the interesting thing is that the GLA domains of prothrombin and 10A align with the C1 and C2 domains of factor 5. So they form this membrane binding platform that is responsible for the activation. So the other important thing is that 10A, both 10A and prothrombin assume this curved conformation. They're not elongated as predicted by computer modeling of this complex. So the cryo-EM structures revealed a completely different conformation that was confirmed by independent measurements with SM-FRET in this case. And then what's important is that we could see very clearly that arginine 320 was presented to the active site of 10A, and arginine 271 would sit about 20 angstroms away. So this complex basically validates the biochemistry that says if you carry out this reaction in vitro, you would see that the preferred site of cleavage would be arginine 320, and that's indeed what we see here in the cryo-EM structure. And so this is a view of the complex where 5A from the top, where 5A is in surface representation, and then you have this cartoon representations for prothrombin, and for 10A, you have the arginine 320 here in green that penetrates the active site of 10A, and the other sites of cleavage, arginine 271, sits on the opposite sides of the complex. And you see very clearly, okay, that basically the gate, what we call the gate in blue here, has the function to sequester arginine 271, putting it away from the active site of 10A, and then directing the rest of the A chain to point to the arginine 320 into the active site, ready for cleavage. Some of the regions of prothrombin and some biochemistry studies said that were very important for the interaction, like the exosite 1, don't seem to make any significant contacts. What's important, what really drives this interaction is the A chain of prothrombin. And this is in nice agreement with the clinical evidence that most of the mutations that affect the activation of prothrombin in the patient populations are mutations of the A chain. So the structure really does justice not only to the biochemistry, but also to the clinical observations. And so basically the mechanism for prothrombin activation, according to the structure, is that the lid here drops on the protease domain of 10A, oriented in such a way that can now interact with the arginine 320. The gate sequesters arginine 271, put on the other side, and drives the rest of the A chain into the active site of 10A to present arginine 320, and that the A chain of prothrombin is the major determinant of interaction. And more recently, this is not published yet, we have been able to solve also the complex of prothrombinase with mesothrombin. So in the top here, you have the complex I just showed you between 5A, 10A, and prothrombin. And underneath here, you see the same complex of 5A, 10A with mesothrombin. And for this complex, we could not resolve the GF1 and GA domains of 10A due to disorder. That's not unexpected. But again, what we see is that the major driving of the interaction between the enzyme and the substrate is the protease domain. So everything happens here with the protease domain, and then the rest of the molecule, as you can see here for prothrombin, rests separated from each other. So what determines the specificity are very few residues located in the protease domain. And I want to show you here a movie that shows this drastic rearrangement that brings the arginine 271, this one, from the position it has in prothrombin up here. If I can go back. To down into the active site of 10A. So this is a massive rearrangement where the 271 is driven by not a conformation of transition, but a rotation of the protease domain to bring the second site of cleavage into the active site of 10A. And basically gives us a very nice structural depiction of the entire activation pathway of prothrombin along the mesothrombin pathway. And as these data are not published yet, are under review. So I want to conclude by saying that absolutely cryo-EM is the way to go to study coagulation factors and their complexes. For factor 5 and 5A, the B domain is no question is the key to understand the function of factor 5, but it's still unresolved. We're getting there. The 5A selects the pathway for prothrombin activation, and there is no question that 5A also acts as a scaffold that organizes the position of both 10A, prothrombin, and mesothrombin. And then the last thing that I want to point out, for those of you who use AlphaFold, when you use AlphaFold, don't forget about the post-translational modification. If you use them, you can make predictions that will surprise you, will be very, very different. Of course, if there are post-translational modifications, but we were very surprised to find completely different predictions for the structure of factor 5, and especially the B domain. And so these are the people in my lab that have done the work. I talked about Kevin, done all the work on the model of the B domain. The cryo-EM structures that I presented are all solved by Eliza and Bassem. Bassem is now an assistant professor in my department. Eliza works at the University of Texas at Austin. And I want to acknowledge Catherine Bazor at Washington University, who's helping us with the cryo-EM. And of course, the NIH, who continues to fund our work after more than 30 years, and hopefully will do that until I retire. Thank you so much for your attention. I'll be more than happy to take any questions.
Thank you, Enrico. Yes, sure. Professor Salvatore.
I just have a curiosity. It's a beautiful work and beautiful images you have shown. I wondered a few things very maybe a little naive. To choose EM as in comparison with the crystallographer, it's because crystallography is not so efficient in this case. How about the resolution at the atomic level of the, how many angstrom is the resolution? And the third thing I want to ask you is the post-translational modification. And the AlphaFold, I don't know if I gathered well, allows you to understand better if post-translational modification of protein are present, and how about the physiology of these post-translational, because I am rather interested in aging, what happens in aging, and if they affect the alteration at the level of the protein. If these post-translational modification, they affect in the sense of pathological status, and has been found something that explains the importance of these post-translational modification, because particularly the glyco ones are very different one from another. And what alpha fold methodology, computer methodology, could give in help to understand exactly these things. Sorry, I made a little bit
That's wonderful. These are great questions. So I guess let's go in order. First of all, X-ray. Why use cryo-EM instead of X-ray? Well, it's not that we haven't tried. We and other people have tried for many years, for 4 decades, to crystallize some of these proteins by X-ray, and some of them are just impossible to crystallize, just impossible. And they're not just the complexes. The complexes have been, the only complex that have been solved by X-ray is the tissue factor 7A complex, which I'm told is very easy to solve. But other complexes have not been solved by X-ray. And also individual proteins like protein S, protein C, there is no structure available. And that's because they have disordered regions and they're just not amenable to X-ray crystallography. Now, cryo-EM has been able to solve some of the proteins, but to get back to your question, you don't get the resolution that you get with X-ray. So cryo-EM will give you an answer, but in some cases the resolution would be 3.2, 3.8. Sometimes it could be even above 4. But nonetheless, it really depends on the questions you ask. If you want to know how a large complex is assembled, which protein binds where, cryo-EM is the perfect way to go. If you're interested in atomic details of an interface, I don't think you will ever get that, not at the current resolution of cryo-EM microscope. And the questions of the post...
You can get up, down, I don't know now.
You can go
1.5 angstrom.
Oh, even below that. It depends on the protein. I mean, we get structures of thrombin now, 1.3, 1.2, which 30 years ago was unthinkable.
Yes.
Okay, so I would say maybe 10 years from now, you'll be able to get cryo-EM structures typically in the range below 2 angstrom. But right now, that's very rare. The typical resolution for a cryo-EM structure is high 2s, mid 3s, if they're really good structures, okay? For the glycosylations, you're absolutely correct. I mean, that's very important. It's a very important issue, so it's important to know what residues are glycosylated, if that changes with age, what's the program, what's the effect on the functionality of the protein. So the data set that Kevin used to build the AlphaFold was basically using all the sites of glycosylation. That may not be correct. Now maybe that's why you get this superstructure. But if you're specifically interested, say, in the glycosylation of a particular residue in your protein, and you want to predict what that structure would be when you glycosylate that site, I really suggest you use AlphaFold and then introduce the sites of glycosylation in the place you want and see if the structure changes in a way that explains the pathology that you observe. But I definitely recommend that you use
Simulation.
Yes, yeah, but you can solve the structure of the, depending on the protein. I mean, you will never see glycosylations by X-ray, that's for sure. But with cryo-EM, you will. With cryo-EM, you can see blobs that are really conducive yeah, that are conducive to glycosylation. We see this regularly. We've seen it with APC, with other proteins, and in fact, use those as anchor points to tell, okay, this must be this domain and this section of the domain because I see a blob here, which can only be a sugar. So that's a very, very important point.
Other questions also from people online? If you want, you can. Luigi Vitagliano, please.
Can you hear me?
Modification, for instance, along with the
Yes, sorry, just 1 minute.
Yeah, of course.
Yeah, I'm sure there are those data, maybe even for factor 5. But as I said, one will have to look at the database and see which site changes the glycosylation, whether there are additional sites that are glycosylated as you age. So that's not something that we have, but it's a very important point. You're absolutely correct. That gives us an idea to look at the progression, if it's known in some cases, whether that would change the structure. Thank you.
Luigi, please.
Okay. Very brilliant presentation. I have some curiosities, if you are hearing me. The first one is related to prediction of protein S. Did you check, does AlphaFold always predict the elongated shape, or you have solutions by AlphaFold changing the seed of fold or looking into different models in which it's contracted? That's the first one.
Yeah, that's a great question. The AlphaFold, first of all, I say the confidence is not very high. The confidence is never high when you don't know the structure. So you should accept that prediction with a grain of salt. But one thing I didn't mention is that model is one of many. So we have several predicted models of AlphaFold. And some of this model, for example, predicts that the B domain will go all the way down under the C domains, which will not be consistent with the biochemistry because it will prevent factor V from binding to membranes. So we just selected a model that will be in reasonable agreement with what we know in terms of the biochemistry. But you're absolutely correct. With AlphaFold, you don't get a unique model. You always have a very low confidence. And so things can change depending on which model you pick. And what we're doing now is introducing what's known in terms of the natural occurring mutations of factor V and see whether they map in regions of the B domain that will make sense in terms of generating a pathological condition.
And the second question is about the B domain prediction. Have you tried to do it alone without the rest of the protein? With AlphaFold, I mean?
No, the AlphaFold, the prediction was done with the entire protein.
Yes, of course. I was wondering whether this superstructure could have been seen also when the B domain is alone, and if this superstructure has some structural analogs in the PDB, at least for fragments or something like that.
There is absolutely, the weird thing about the B domain, it has no sequence homology with anything that exists on this planet, except this very brief segment of TFPI alpha that is homologous to the to the basic region. In terms of anything analogous in the PDB, there is nothing analogous. But Leslie, the person I have in the lab, who's been my lifelong technician, recently succeeded in expressing the B domain alone. And we're very excited about that because I think we can solve the structure of the B domain alone. And that, of course, doesn't guarantee that the structure will be the same as the structure of the B domain attached to the rest of factor V, but it for sure will tell us whether it has a superstructure or remains very disordered. So we're going after this B domain alone that apparently nobody's been able to obtain for many, many decades, starting to work on the protein, and we'll see what happens. So stay tuned, we'll find out.
And have you tried predictions for the acidic region with interaction with the TFPI alpha? I mean, your suggestion has been tested with AlphaFold as well. I mean, the one that you're working on, if the
I'm sorry.
now the interaction of the acidic region, the red region you have on the protein, and interaction with the inhibitor. I was asking whether this, and you are working on the modeling of this interaction as a.
Yeah, we also have preliminary data
Yeah.
of that's
Yeah.
the fragment of the FBI alpha bound to 5 short, because there you will have the interaction. So we
Yeah,
made a
but I was
peptide
asking-
of the FBI alpha, the basic region, interacting with 5 short. We got preliminary data of that. We see something, not very clearly, but there is something attached to the acidic region.
What about AlphaFold in this interaction? Have you tried predictions? to see-
As I said before, depends on which model you pick. But in
Okay.
most of the models, you see the BR and the R coming together. Even the spaghetti, like completely disordered, they don't come close together for interaction, but they come, they come toward each other. So that there is an electrostatic interaction, it makes sense. But depending on the model you pick, the actual interface between the BR and the AR changes. And of course, you can make mutations to test the various models, but working with factor 5 is
Yeah.
very difficult. So you don't want to, it's not like a trypsin, you can make 25 mutants and select the ones. I mean,
Okay.
you have to be very certain that you're after something. Otherwise, spend a lot of time and efforts for nothing.
Okay, thank you.
Thank you.
Other questions?
Posso chiedere una cosa che non c'entra?
Sì,
Yes,
ti do una risposta che non c'entra.
Io non sono un biochimico, per cui mi interesso, cioè sono un pediatra prestato alla genetica medica, ma durante l'embriogenesi la sintesi dei fattori della coagulazione è sincrona, vale a dire compaiono tutti allo stesso momento. Questo perché alcuni dei fattori hanno dei fattori trascrizionali specifici, tipo il secondo, il settimo, anche il quinto, per cui si potrebbe pensare che alcuni compaiono prima, alcuni compaiono dopo.
È una domanda bellissima. Io non sono un genetista, quindi mi devi perdonare per un po' l'ignoranza. Quello che so è che il primo fattore della coagulazione che compare durante l'embriogenesi è la protrombina. E il motivo per cui la protrombina compare prima è perché originariamente non era un fattore della coagulazione, ma era un growth hormone. E infatti la protrombina nell'embrione la trovi prima di tutto nel cervello, e nessuno è riuscito mai a capire perché si esprime la protrombina nel cervello, perché effettivamente, cioè, si ricorda del suo ruolo originario di growth hormone, la trovi là, poi dopo viene sequestrata, sono tutte espresse dal fegato, non entra più, non passa più la barriera per entrare nel sistema nervoso, e funziona semplicemente nella coagulazione. Un altro fattore che compare abbastanza presto è il tissue factor, che è praticamente il fattore che ti triggera la coagulazione. Quindi in effetti i fattori che compaiono prima sono i fattori più importanti, quelli che eliminati sono fatal, cioè non sopravvivi, e sono appunto la protrombina, la proteina C, il tissue factor. Gli altri invece sono veramente disposable, quindi adesso non so esattamente la cronologia, ma so di certo che non sono i primi a comparire. Quelli che compaiono per primi sono quelli che devono trigger la coagulazione, hanno altri poteri come la protrombina di agire come growth hormone, come ormoni della crescita, e poi la proteina C che controlla, che si assicura che la coagulazione, quando è attivata, viene terminata.
Altre domande? Posso chiedere io una cosa?
Certo.
Vabbè, innanzitutto volevo capire questo dominio B se è stabile, aggrega o rimane in soluzione. E la seconda cosa è se questi studi possono portare al disegno di inibitori specifici, un po' come ci sono per il fattore decimo o la trombina, se pensi che possa essere un target più...
Il fattore B come target lo vedo male, perché
No, il
proprio
fattore quinto, dico.
il...
Da questi tuoi studi, diciamo, sul fattore quinto in generale, dei punti specifici.
Sì, secondo me è difficile perché a differenza degli enzimi
Che hanno il sito attivo.
dove è più facile definire delle molecole idrofobiche che si legano e non si staccano subito, diventa molto difficile, anche perché le superfici di interazione sono poi limitate e sono tendenzialmente elettrostatiche. E come sai, interazioni elettrostatiche in acqua non è che sono molto forti. Cioè, quindi non vedo il fattore quinto come un target per la mutagenesi o per lo sviluppo di inibitori. Per quanto riguarda il dominio B isolato, si aggrega, quello che stiamo vedendo con Leslie che si comporta perfettamente stabile, non aggrega, viene però mangiato facilmente da altre proteasi, quindi quello è un problema di stabilità, perché è talmente esteso come dominio che bisogna stare attenti a inibire parecchie proteasi che stanno in giro.
Ok, grazie. Altre domande? Mena.
Grazie, una bellissima presentazione, fantastica. Richiamo una cosa di cui non hai parlato. Nel passato voi avete studiato e avete dimostrato l'esistenza di un equilibrio conformazionale in soluzione della protrombina tra una struttura chiusa ed una struttura aperta. Nel complesso con la protrombinasi, diciamo, è presente solo la struttura chiusa della protrombina. Lo stesso, se capisco bene, rimane anche nel nuovo complesso, quello con la mesotrombina?
No,
No.
purtroppo la mesotrombina che abbiamo usato è la DSF1 mesotrombi, quindi è la mesotrombina che non ha né il gladomain né l'IGF1. Noi abbiamo cercato di cristallizzare la mesotrombina, con la protrombinasi non riuscivamo mai a ottenere dei buoni risultati in termini di risoluzione. Quindi abbiamo sostituito la mesotrombin col DES1.
Okay.
E quindi non avendo il gladomain
non sappiamo qual è la conformazione,
esatto.
Quindi è ancora...
esatto, questo è un problema che
non è stato ancora risolto.
Non abbiamo potuto risolvere con la struttura, purtroppo. Quello che abbiamo voluto vedere è come interagiva il protease domain con il resto della protrombinasi, ma purtroppo non sappiamo dove sta il gladomain o l'IGF1. Quindi
vabbè, in futuro va.
sì, vediamo se qualcuno ha la pazienza di andarla a vedere. Ma tu hai perfettamente ragione, l'equilibrio importante è per la protrombina, è confermato che la forma che si lega è quella closed.
Ok, grazie.
Allora, grazie ancora. Se non ci
Grazie a
sono
voi.
altre domande, ringraziamo Enrico. Arrivederci.
Grazie a tutti, grazie. Grazie per la domanda, bellissimo.